In the formation of disaccharide, two monosaccharides are held together by a bond called glycosidic linkage. Now take some example of disaccharide. One disaccharide is called maltose, which consists of two molecules of glucose. This is one glucose. This is one glucose. Now one more thing, if at carbon number 1 hydroxyl group is written below, it is designated as alpha form. So in both glucose molecule, hydroxyl group is written below. So both are alpha glucose. Now two glucose are linked by 1,4 glycosidic linkage. In the formation of glycosidic linkage, one molecule of water is removed. So hydro bond is formed between carbon number 1 of one glucose and carbon number 4 of other glucose. That's why it is called as 1,4 glycosidic linkage. And since the linkage is in the glucose involved in formation of maltose are alpha glucose, that's why it is called alpha 1,4 glycosidic linkage. Other disaccharide, lactose, which is a constituent of milk, consists of one molecule of galactose and one molecule of glucose held together by beta 1,4 glycosidic linkage. This is structure of galactose. What is distinguishing feature of galactose? Hydroxyl group at carbon number 4 is present above. This is distinguishing feature of a galactose. And this galactose is beta galactose because at carbon number 1 hydroxyl group is written above. Similarly, in this glucose molecule, hydroxyl group is written above. So this is also beta 4 and this is also beta 4. That's why linkage is beta 1,4. 1,4 means glycosidic bond is formed between carbon number 1 of galactose and 4 of glucose. Sucrose, which is a cane sugar, consists of glucose and fructose. Linkage involved is alpha 1 beta 2 glycosidic linkage. This is alpha form of glucose because hydroxyl group is written below. This is beta form of fructose because at carbon number 2 with the nearest carbon linked to oxygen, which is also called as anomeric carbon, is written above. So linkage is formed between hydroxyl group at carbon number 1 of glucose and hydroxyl group at carbon number 2 of fructose. The linkage therefore called as alpha 1 beta 2 glycosidic linkage. Another group of carbohydrate called polysaccharide is further of two types, homopolysaccharide and heteropolysaccharide. Homopolysaccharide on hydrolysis yields similar type of sugar in it. It yields dissimilar type of sugar in it. Some of the examples of homopolysaccharides are starch, which is a glucan. The polysaccharide of glucose is called glucan. So starch is a glucan. It consists of many molecules of glucose, which are held together by 1,4 as well as 1,6 glycosidic linkage. In a straight chain part, linkage is 1,4. At the point of branching, linkage is 1,6. Again, in a straight chain part, linkage is 1,4. So 1,6 linkage is only present at the point of branching. Otherwise, linkage is 1,4. It is starch is storage polysaccharide of plants. Other homopolysaccharide is glycogen, which is also a glucan. It is often called as animal starch. It is stored in our body, in liver as well as muscles. Like starch, it also contains 1,4 glycosidic linkage in straight chain part and 1,6 glycosidic linkage at the point of branching. Cellulose, which is most abundant natural polymer because it is essential constituent of plant cell wall. It's a structural polysaccharide. Linkage involved is beta 1,4 linkage. Inulin, which is polysaccharide of fructose, that's why it is called fructan, is often called as dahlia starch. Dahlia starch. Some of the important example of heteropolysaccharide, which is dissimilar type of sugar units, is hemicellulose, which contains xylose, arabinose, and other types of sugar units. Hemicellulose is found in association with cellulose in plant cell wall. Similarly, mucopolysaccharide is also example of heteropolysaccharide. It contains many types of sugar units. Essentially, it may contain acidic sugars, as well as amino sugars.